Good afternoon, everybody. The so-called GY 971 is our new molecule that is a new derivative of a new class of angular furocoumarins structurally related to TMA, the 3-methyl-angialisine that we study for several years. And it is known to be an anti-inflammatory agent and also a corrector of CFTR, but if used at high concentrations and under UV radiation, possess some side effect. For this reason, we studied a lot of new generation TMA derivatives synthesized and characterized by Adriana Quilin and her team at the University of Padua. And through both in vitro and in vivo experiment, we identify GY 971 that you can see possessed the same skeleton, angular skeleton of TMA, but it is characterized by a new bulky moiety in position 6 that avoids all the mentioned side effect typical of TMA. Actually, we utilize for all the experiment of this project it's mesylate salt, GY 971A, actually a little bit more water soluble than the free amine, and that we validate, verify not phototoxic and not genotoxic. Our major goal of this concluded project 2022 was the validation of GY 971A, that for brevity I'll call simply GY, as potential anti-inflammatory agent, and at the same time studying possible side effects. In order to do that, we were supported by two essential services of our foundation, the service of primary cells, directed by Valeria Capurro at the Gaslini Institute with Nicoletta Pedemonte, that gave us all the sample, all the bronchial epithelial primary cells derived from people with cystic fibrosis. And also the important CFA core service directed by Alessandra Bragonzi for all the in vivo experiment utilizing murine models, different murine models. The preliminary experiment were done on cystic fibrosis bronchial epithelial cell lines, such as CFBE, actually in order first to understand the anti-inflammatory activity of GY. And GY doesn't affect the cell growth at nanomolar concentration between 15 to 200 nanomolar and apoptosis 2. And was able to reduce the interleukin gene expression and production in this cell line. The interleukin gene that is a biomarker, non-biomarker of CF inflammation. And the first in vivo model was a murine model of acute infection caused by Pseudomonas aeruginosa. In this mice, GY was administered by a pan-century mediated aerosol system, because our idea is the possibility to have a good delivery system by aerosol for pulmonary delivery. GY induced in this model a strong decrease of inflammatory cells, total cells, including neutrophils and macrophages too, and at the same time without increasing the bacterial load in murine bulk. All the ex vivo studies of the anti-inflammatory activity of GY were conducted in our labs, particularly by Chiara Tupini and Sofia Fagnani in collaboration with Anna Tammannini. We utilize all these human primary bronchial epithelial cells first to understand, to check the viability of our derivative. I didn't determine antiproliferative activity or pro-apoptotic effect in these primary cells. Okay. We were focused on the method setting in order to obtain a good primary cells model derived from people with cystic fibrosis carrying the F508 deletion. We utilize cells, primary cells, without any stimuli, and also utilizing pro-inflammatory challenges, including Pseudomonas aeruginosa, different dilution, or its secretome. Thank you, Giulio. Its secretome, different dilution, and also two characteristic cytokines, TNF-alpha and interleukin 17A, alone and in combination. As you can see, all these stimuli were capable to increase the interleukin 8 expression, actually upset interleukin 70A at different concentration, and these data were confirmed also at the protein level, measuring in the basolateral or apical media in the transwells on the protein. And lastly, we have chosen two of these stimuli, the Pseudomonas aeruginosa 1-1 and TNF-alpha at 5 nanogram ml, that in our experiment were the stimuli that induce pro-inflammatory effect in a more stable way. So the effect of GY at different very low concentration, 10, 50, and 200 nanomolar, were verified on the constitutive expression not only of interleukin 8 chemokine, but also on the expression of TNF-alpha, interleukin 1 beta, and the interleukin 6 genes. As you can see here, the result from different epithelial derived from different people with cystic fibrosis demonstrated that GY was able to reduce the transcription of especially interleukin 8, TNF-alpha, and interleukin 1 beta. Also in the model of these primary cells, TNF-alpha stimulated, the expression of interleukin 8, TNF-alpha, interleukin 1 beta, and interleukin 6 gene were decreased after treatment. And the more relevant result were observed when primary cells were stimulated with the Pseudomonas aeruginosa. In this case, we can observe a very interesting modulation of GY at different concentration in a dose-dependent manner on all the chemokines and cytokines involved in inflammation, in CF inflammation. So we know that GY is able to modulate genes of pro-inflammatory molecules, and we know also that some author published that inflammation increase the rescue of F508 deletion protein by ETI. To be sure that GY doesn't interfere on this important rescue, Western blot analyses were carried out in our lab. You can observe that in all lane there is the B band, and the C band appears when ETI, we treated the cells with ETI, and also when ETI plus GY at different concentration, without inflammation, without Pseudomonas aeruginosa infection, and also with infection. And these results were verified also in primary cells, and also in this case we can observe that without and with Pseudomonas aeruginosa infection, the C band is present and our derivative did not interfere on the rescue. Okay, lastly, to exclude the possibility that GY promote possible systemic effect, stopping the acute inflammatories and acute inflammatory response during possible infection determining hepatotoxic effect, we schedule a new experiment, in vivo experiment based on a murine model of acute LPS-induced inflammation, designed and organized by Alessandra Bragonzi and Giulio Cabrini. And we evaluated the hepatotoxicity first with the measure of body weight and body temperature of mice treated with LPS and GY, and also the level of AST and ALT with the same treatment in comparison to a known positive control, the D-galactosamine, that increase with a stronger increase the level of both AST and ALT, or determine death of mice. So, in conclusion, I would like to highlight that a good pro-inflammatory of study was developed in ex vivo, utilizing ex vivo primary cells derived from the biobank of Institute of Genova, Gaslini Institute. This model can be utilized also in the future to test, to study other possible anti-inflammatory molecules or other anti-inflammatory strategies, different strategies. All the utilized stimuli that we used in this particular cells, precious cells, clearly increase the basal level of all the chemokines and cytokines involved in inflammation. We have chosen two in particular stimuli, the Pseudomonas aeruginosa and TNF-alpha, that were able to increase the basal inflammation status, and these primary cells actually responded very well to the treatment with the very low concentration of GY, which was able to reduce both basal and stimulated the transcription of all the chemokines and cytokines genes studied. These results were also confirmed at the protein level, and also in this case, the effect of GY is a variable among these primary culture, these primary cells from different patients, maybe not only because of normal experimental viability, but also, I don't know, for specific extra CFTR genomic background. Not only did we exclude negative interference of GY with the ETI, CAF3 or Trikafta, but we even found some positive synergetic action of GY on ETY, actually, although very limited. And for the future, thanks to the newly funded project 2024, titled GY971 as anti-inflammatory agent 2.0, we will validate GY utilizing again our model, our primary model, but also a new model ex vivo of human nasal epithelia derived from nasal brushing of people with cystic fibrosis. And we will utilize also more mice model, chronic model of infection and inflammation, and if possible also CF murine model. And the new, for us, new model zebrafish. We will start studying the very important for our project optimization of pulmonary delivery methods to deliver in vivo, in chronic experiment, our GY, our new derivative, and we will evaluate also major safety issues, starting with ADME toxicology in silico, potential off-target molecules in vivo, using the 44 target assay, GY971 effect on transcriptomics with RNA-seq experiment, and more standard preliminary toxicology in vivo. We will start with this new project with some preliminary results that we summarize here, but you can read in our poster. For instance, the in silico docking experiment revealed that GY was very similar, but possess an affinity stronger than TMA for the protein pocket P50 of the NFKB transition factors, and we validated also the anti-inflammatory effect of GY in the nasal epithelia and also in zebrafish larvae. For the first project 2022, I would like to thank all the mentioned colleagues, internal and external collaborators, and FFC, and all the delegations from Treviso, Montebelluna, Pesaro, Rivaloro, Canavese, Parma, and Fidenza. And with the new project, we were happy to enlarge the team with the collaboration of Rita Cortesi from university for all the delivery system studies, Marco Prosdocimi, Rare Partners that help us in the orphan drug designation, Onofrio La Selva for all the experiment in the nasal epithelia, Roberta Roncaratti from CNR Bologna for the future transcriptomics analysis, and Marco Cafora with Anna Pistocchi for zebrafish, for the zebrafish model. And the new delegation from Massafra, Treviso, Montebelluna again, La Bottega delle Donne, Torino, Ferrara, and all of you for your kind attention. Sorry for my sick.